Hi, welcome to theme 5C for the PCE3 workshop. Theme today is Rewinding Life's Clock. My name is Greg Fournier. I am a professor of geobiology in the EAPS department at MIT. So comparative phylogenomics is a top-down approach that we can use to understand the earliest events in the history of life on Earth. So as opposed to, say, looking at prebiotic chemistry and coming up with plausible ways that life could originate, and the earliest steps from abiogenesis to organic synthesis to combining complex organic molecules toward lifelike systems, what we do is look at existing living systems, comparing sequence, physiological characteristics, or different properties across existing diversity of life, and then we work backwards, trying to reconstruct more ancient states leading back to the last universal common ancestor and even earlier, reaching towards the prebiotic world. And this is called a top-down approach. So this allows us to ask a lot of interesting questions because unlike prebiotic chemistry, unlike bottom-up approaches, this is inherently a historical process. We are trying to rewind the tape of life and infer events that actually occurred as far back as we can. And so we can think about some of the big questions. What properties can we infer for early life? How far back in time can we go? Do the observations we can infer with a top-down approach, do they converge upon, or do they at least agree, or are they consistent with any of the narratives that have been established by our collaborators working in the field of prebiotic chemistry? So before we talk about how we can use the phylogenomic record working backwards to talk about origins, we should go over some of the basic principles at work here. So the first of these is a very old principle, going all the way back to Darwin. And this is just comparative evolution. When we study evolutionary biology, what we are doing is we are comparing organisms alive today, and we're looking at similarities and differences between them and using those to not only infer relationships, but also to infer what their ancestral states were and the processes that led to the states we observe today. So here is a cladogram, a very simple example of this. And what we see is if we have a table that just lists the different characteristics we observe for organisms alive today, we can use this to infer a parsimonious evolutionary history. And we can see that groups on this tree are joined by derived characters. For example, reptiles and mammals are amniotes. They have an amniotic egg. And so we know that the amnion must have evolved before these groups split in the tree. So we'd say this is a derived character grouping them. The outgroup would be non-amniote tetrapods or amphibians. And so as we're going further and further back in time, we can build up an idea of what the ancestral lineage may have looked like, what characteristics it had, and what characteristics it did not have. Now, this is no different than doing phylogenetic studies with sequence alignment, except for sequence data, we have a much more robust set of information that we can actually make predictions with and quantify in phylogenetic reconstruction. Also, microbes, unfortunately, don't have this nice morphological set of characters or a very deep fossil record. And so sequence information is really all we have if we're trying to reconstruct the deepest splits in the tree of life. So how do we do that? Very, very briefly, what we do is we take aligned sequences, either DNA or protein sequences from organisms alive today, and we can therefore determine what individual sites within their alignment are homologous or share common ancestry. And then we simply look at the changes. What are the transitions between these different states? We apply an evolutionary model, some belief in the probability of expected changes between states. And then we have to ask the question, what is the evolutionary history that best explains the observed distribution of states in our sequences given this model. And if we can do that, we end up with a phylogeny, a phylogenetic tree where the tips represent the species existing today. And as we go backward, they converge to common ancestors deeper and deeper in time until we get to the root of the tree, or the common ancestor of all of the sequences that are present in our data set. So people have done this many times, looking at the entire tree of life and looking at phylogenomic data sets of conserved proteins that are found in archaea, bacteria, and eukaryotes. And this forms the scaffold of a lot of the analyses we're going to talk about today. So this is the tree of life rooting all the way back to the last universal common ancestor of Luca. And when you have this tree, you can ask a lot of really interesting questions. You can compare different gene families across this tree and say, well, where are they present? Where are they absent? What are their histories? Were they likely present in Luca, or did they evolve later on? You can map different properties. For example, that archaea have different membrane chemistry than bacteria. We can map it on the tree and infer on deep branches these changes must have occurred. You can also work backwards and reconstruct what you think the earliest metabolisms were for different groups on the tree. You can also look at the shape of the tree itself and the evolutionary distances represented by branches and apply molecular clocks to try to interpret these genetic distances as units of absolute time to tell us something about how old different groups are in the tree of life, or potentially even how old Luca is. So if we simplify that down and just collapse it to think about these basic concepts, we can have a very simple tree here where we have Luca and then the two major splits leading to bacteria or archaea. And then all the way over here, very early on, we have the origin of life. And you can see how bottom-up investigations like prebiotic chemistry are really, this is their domain, talking about origins. And the top-down comparative genomic approaches, which are historical, reach back to Luca. The in-between is the interesting part, where we don't know what's going on. And so I like to call this the dark age. This is when we still had inheritance. And so this is still a historical evolutionary process leading to Luca. However, because it predates the coalescence of the last common ancestor, we can't use normal comparative genomic methods to infer any of these early events that led to Luca. But if we were able to push this back in time, one would hope that it would start to converge on what is observed through prebiotic chemistry or experimental studies of origins. So we can divide up these intervals of time into three pieces. Number one, when we have the origins of all the inherited genotypes and phenotypes that we observe in Luca. Number two, Luca itself. What was the nature of Luca and life at the time of Luca? And then three, just after Luca, the evolution and diversification of the major domains. So here we're well within standard comparative genomic analysis. So we can think of these as three different ages of life. So the first stage is what I call the dark age. So this is just the origin of life until first heredity. This is dominated by prebiotic chemistry and chemical evolution. We have no physical record. And this may be very brief. These processes may have occurred on subgeological timescales, or they may have occurred independently in many different places on Earth. The second age is really this age of universal ancestry. That is when you first start to see genetic material that is passed down, the rules of natural selection in place, and inheritance that has persisted until today. So the origin of all the genetic material that was inherited by Luca and subsequently passed down to life that we can observe in sequence now. And then finally, the third age is just modern life. It's everything after Luca, what we normally think of as life. And this is the source of a lot of the information that we use working backwards to try to infer the processes in earlier times. And once again, the big question is there a process continuity? Can reconstructing the states of genetic components and molecular biology that exists today, does it produce a reliable arrow that can infer what was going on in these very earliest stages? Or is there discontinuity? Was there takeover from completely different systems? And therefore, investigating top-down approaches can tell us nothing about the intermediate steps. And that's also a possibility that we need to seriously consider. So in thinking about LUCA specifically, this process of coalescence becomes very important. So as I mentioned, we can work back in time from all of these observed tips. And in this tree, we see the black represents ancestral lineages to extant forms. In the gray are all of the extinct branches on the tree of life. And if you go back in time far enough, you reach a sen ancestor, or the last common ancestor of all groups alive today. But you can see there's no reason to believe that LUCA was alone or the first organism. It's just the last common ancestor that survived. There may have been many other branches present at the time of LUCA. They may have had many descendants, but for whatever reason, they did not survive until today. So when we think about LUCA, we're not necessarily thinking about a singular origin. We are thinking about just the common ancestor of existing life that we can infer back to. And then we have this idea of FUCA then. What is the first universal common ancestor, the true origin of cellular life? And that is a very different question than what we can ask about the last universal common ancestor. So what do we know about LUCA? Well, in some ways we know a lot, and in some ways we know very little. Just from comparative genetics, looking at different groups on the tree of life, it's very clear that LUCA had more or less a complete genetic code, a complete ribosomal machinery for synthesizing proteins, the ability to polymerize RNA, and it was some kind of a membrane-bound organism, although we don't know what the chemical nature of that membrane was. But there are many commonalities between different domains and many differences that can inform what is likely ancestral and what is derived with relationship to LUCA. But it seems very clear that there was a universal genetic code, protein translation machinery, the ability to make RNA, and likely an ATP-based energy metabolism. Beyond that, we could say that it's likely that there was DNA, cellularity of some kind, and then a conserved core of cofactors for metabolism and intermediate metabolic enzymes. The things we're not too sure about are chemistry of lipid membranes, because that differs between archaea and bacteria. What the particular energy metabolism was. I mean, we can't really work directly back to that. We have to apply hypotheses as to what available sources of free energy would be to an organism like LUCA. And where LUCA existed under what environment is also largely unknown. The genetic code is probably the most complex and really definitive character that we can root back to the time of LUCA. And if you have a genetic code, a syntax in place for translating DNA sequences into functional polypeptides, you must have tRNA. You have to decode this. You must have synthetases for loading the tRNA with cognate amino acids. You must have the ability to modify tRNA chemically in order to produce all of the anticodons needed to partition up the code in this complex way. Now, we don't really know how the genetic code originated and evolved. There are many different hypotheses that are consistent with different particular features of the code. A nice review of that is in Trifonov from 2000. And they have some commonalities. It seems clear that very similar amino acids in the code are encoded by similar sets of codons that may suggest a shared ancestry or a process by which new amino acids were added to the code, or by which specificity was increased over time. But we don't know. And the further back in time you go, the more and more speculative this gets. But one thing that I think is absolutely fascinating is the code did not continue to evolve in any really important way after LUCA. Archaea and bacteria do not use radically different sets of amino acids or codons. So regardless of the selection and the evolutionary process that led to the genetic code, it was complete by the time of LUCA. And there was never sufficient selective pressure after LUCA to radically revise this going forward, even given the incredible explosion of diversity of life on Earth. And we don't know why that is, but I think that's one of the major outstanding questions. We can also look at inferring states of LUCA beyond simple sequence data. For example, we can look at and compare structures. So some work out of Lauren Williams' group has done this for RNA, and found that in highly conserved ribosomal RNA, there are these little pieces that are universally conserved across life. And there are other parts that seem to elaborate as you look at more and more derived groups of organisms. So you can use that to kind of work backwards and infer that there's been a complexification of these states, and that the little pieces that were conserved must be molecular fossils representing some kind of a state that was present at LUCA or earlier. You can also think even bigger scale to the ribosome itself. So this has been called the onion layer model of the ribosome. The idea that you have this big collection of proteins and RNA working together, and if this must have accumulated over time. And so if you work backwards and go deeper and deeper in time, you should come to a simpler system that is represented by components that are closer and closer to the catalytic site. And so you can, when you peel away these layers of the ribosome, you're going backward in time, and that can tell us something about the early history of translation. One major outstanding question about LUCA is, what is its relationship to cellularity, and what is its relationship to inheritance, to just the rules of Darwinian inheritance? And there are two major hypotheses for this. One is the LUCA late model, where LUCA is really just a cellular lineage like anything else. There were many other cellular lineages at the time that did not survive. And the very early stages of life that may have preceded Darwinian inheritance and cellularity belong to some distant age before LUCA. Alternatively, there's a LUCA early model where this progenote, or this pro-Darwinian living system, really is synonymous with LUCA, and cellularization and kind of reaching this Darwinian threshold occurred independently in lineages leading to archaea and bacteria. And so that's another set of questions that have yet to be completely resolved. What genes were present at LUCA? So in a very simplistic way, you can simply look at all the genes that are universally conserved and assume that these were present in the common ancestor. But this might not be true, because gene transfer could have spread these genes around after the time of LUCA. Conversely, many genes may have been lost in some lineages, therefore inferring false negatives, thinking the gene may not be at LUCA while it is. That being said, this is inherently a question that is going to have an unknowable component, because we know that gene families are lost within lineages today. The same thing must have been true early in the history of life. And so this presence-absence or comparative model for reconstructing what genes are in and not in LUCA is useful, but also has very clear limitations. So what if we push back before LUCA? So can we infer the presence of lineages that diversified before LUCA? Maybe we can, if these transferred genes to groups that are existing today, and that phylogenetic signal may be detectable. A little more tractable is looking at gene histories that diverge before LUCA. So it turns out that if ancient protein families underwent duplications or diversified before LUCA, we can see this signal in phylogenetic trees and can therefore reach back to before that time. And we call these deep paralogs, and these are the earliest information we have using a top-down approach. There are many protein families that very clearly diversified before LUCA. For example, protein families like FITZ that are involved in septum formation and cell division, or the catalytic and non-catalytic subunits of ATP synthases. These make the two parts of the rotary motor that generate ATP from proton gradients. And this is interesting because if we have rotary motor ATP synthases before LUCA, then a direct inference from that is that you also have lipid bilayer membranes at the time of LUCA. Once again, aminoacyl tRNA synthetases, or AARSs. There are two major classes of these that are not homologous. Each class encodes about 10 different synthetases, and all of the divergences leading to the different synthetase types all occurred before Luca. So for example, this tree we have on the left, every tip we have represents a different state that was present at Luca. So all these internal branches must have diverged from each other before Luca, because Luca inherited every one of these versions. And we can see that just looking at the synthetase classes themselves asks another question. They don't show any homology in the terms of sequence. However, they perform very similar chemical catalysis, and they have nicely partitioned up the 20 amino acids into two different types, or two different sets that are amino isolated by either class 1 or class 2. But their differences, they actually chemically work in a very different way on the tRNA. So is this an example of convergent evolution? This kind of shows that pre-Luca, there may have been many independent solutions to different evolutionary questions, and sometimes both solutions were inherited by all life today, but other times maybe only one was, and if the other went extinct, how would we ever know? So what does all of this tell us about habitability or the origins of life? And it's really hard to say anything in terms of definitive statements, but it's better still to think in terms of questions. So how much modern cellular evolution occurred and existed before Luca? Are these deep patterns the result of any bottlenecks or mass extinctions? Or are we only seeing diversification and patterns of speciation and extinction being played out over billions of years? How can we map these early events in the history of life to the notion of a biosphere? At what point are we talking about life being diverse and abundant versus just a unique quirk of chemistry in some vent or in some pool somewhere? And you can even think astrobiologically about these things. How pre-selected are the building blocks of biology given the prebiotic conditions? If they're strongly dependent on prebiotic conditions and these conditions are abundant everywhere, then maybe if life evolves anywhere, it ends up looking biochemically very similar. However, if life has the ability to rewrite the rules, so to speak, using Darwinian selection, there may be a discontinuity and there may be very little in common with how life does biochemistry today versus how it originated, in which case the life would be liberated by the deterministic conditions of its origins. And we don't know which of those is a more plausible scenario. So finally, I wanted to talk a bit about molecular clocks and ancestral sequence reconstruction, kind of two specific applications of gene sequences and phylogenetics. So the length of branches in your phylogeny are a special kind of information that relates evolutionary distance. And if you have some idea of the rate at which groups are evolving, you can simply directly calculate how much time has passed across these branches using a molecular clock approach. It's not as simple as that, but it gives us a nice way to compare events in evolutionary history to events in planetary history. So very briefly, the different components to this are you need a set of calibrations that are constraining the relative or the absolute divergence times of groups on your tree. You need an evolutionary rate model that has a distribution of what you think rates should be on different branches to convert these relative distances to relative times. And you also need to calculate a posterior. How likely is these models, how likely are these models to produce the observed sequence data you see? And you can use that to calculate a probability distribution for different ages for different splits on your tree. But it turns out applying this is very tricky when we talk about origins. There's no outgroup for rooting Luca, and the very deep branches we're looking at are often very long. And we don't know how fast they were evolving. We also have basically no fossils this old, so we don't have a lot of hard ways to constrain absolute times. And it turns out, unfortunately, that a lot of these pre-Luca genes have very fast rates of evolution. because they involve functional divergences, or maybe overall evolution happened faster back then because there was more available sequence space to explore. Um, and so also we run into this problem of just inherent uncertainty. You lose information as you go back in time, and the rate models are probabilistic. And so you may have very different ages that all have similar likelihoods. And the error bars on these ages may be 500,000,000 years and can be just as large as our prior belief in potential age ranges for Luca, for example. And therefore, they may contribute very little to the conversation. However, the most important thing they can contribute is more reliable, younger bounds on groups on the tree of life, like bacteria and archaea, that therefore constrain the window in which Luca and origins must have occurred. Um, so just to show how tricky this can be, here's some work that was done last year that shows that different data sets, looking at different proteins, if you just give a relaxed molecular clock model with no absolute age constraints, because of these very deep, long branches in our tree between bacteria and archaea, these can force the origin of life and Luca to be potentially over 7 billion years old, which we don't think is true. So evolutionary rates and models start to dominate over the data or any calibrations. So we have to be very careful for applying molecular clocks deeper than the constraints we have within archaea and bacteria. It's even worse if we look at individual pre-Luca genes. So looking at our friends, the synthetases again, if we take a molecular clock analysis where we infer that all of their major synthetase families existed at the time of Luca, then we can constrain a molecular clock model by the age of the Earth and see how old these splits are that led to the families. And we can calculate the rates. And if we do that, their rates before Luca are 5 to 15 times faster than their rates after Luca. This variance in rates is so large, we can say next to nothing about these time constraints. And finally, just a little about ancestral sequence reconstruction. This is taking sequence data, taking your tree, working backwards, and coming up with the likely ancestral states for deeper branches, including last common ancestor. And once again, this is very probabilistic. You lose information as you go backwards in time, and you may only be able to say something very, very vague about what the likely ancestor sequence was. So inferring specific hypotheses for the function of ancestrally resurrected proteins is often very difficult, but we can think broader. For example, overall composition. If you look at the overall composition of protein sequences inferred to be present at Luca, for example, we can see that they resemble the composition of proteins known for mesophiles, not thermophiles. And this is an independent way that phylogenomic inference can be used to constrain the environment that Luca likely lived in. Based on these reconstructions, it's unlikely that Luca was a thermophile. We can also use this to investigate the genetic code. If you take a tree of synthetase proteins and you think you know where the root is, you can reconstruct the likely ancestor sequences. And you can look at the presence or absence of specific amino acids. And if we do this, what we can see is all of the 20 canonical amino acids are present with a high probability in at least one site, which tells us that the genetic code must have been complete even before the synthetase families diversified. And this opens up an avenue to ask, well, what was doing the amino acylation? Was it RNA proteins? We have no direct evidence of RNA world amino acylation, but this would infer that something like that may be necessary in the earliest steps of the origin of translation. Thank you for listening to this very brief overview. I can just leave this conclusion slide up here. The single takeaway that I think is the most important is top-down studies are incredibly important for understanding the historical record of life, but very quickly we have to be careful with how we apply this information when we get to the deepest splits in the tree of life, because we have less and less information as we go backward. Thank you.